Okay, let's check for benzene whether it is an aromatic compound or not. Is it ringed? Yes, it has rings. How many pi electrons does it have? Count here. How many pi electrons? 6. Can everybody see 6 pi electrons? And are they delocalized? So this is what we generally study in resonance, which I do not have the time to go over here, but look into this aromatic compound thing if you are interested. What this means is these all electrons are involved in something very special. These electrons are moving around. And that is why like this comes to this comes here and this. So there is a delocalization that is happening. And when all these three things happen together, we call the compound as aromatic, and this brings in stability to the molecule. For example, this is aromatic. This thing also has 6 electrons which satisfy 4n plus 2 pi when n is 1. This thing also has delocalized electrons because there is this alternate double, single, double, single, double. So this is what is needed for resonance. So you need that alternate things happening. It's also happening here, but this is not ringed. What ring does is brings back this electron. Once it reaches here, cannot jump back. But if you make it a ring, this entire thing can actually circle back. That is required for aromaticity. In here, all these 6 electrons are involved in making this compound aromatic. Okay? That is why these electrons are not so much available for reaction. But now take a look into pyridine. What is happening is the structure is very similar to benzene, but in here I want you guys to tell me whether this nitrogen lone pair is involved in making this compound aromatic or not involved in making this. It is not involved. But in some case, you might have electrons involved like in this, but in this case, these 2 electrons are not involved. So these 2 electrons are free to react. That is why this can act as a base. This is the seat of that reactivity. For example, if you have this compound, the nitrogen lone pair in this case is involved in making this ring aromatic or this compound aromatic. So this will not act as a base, but this will. And we will see this thing come many a times in these nucleobases. Always think about that this is a thing that you'll need to understand. Okay, so coming back to pyridine, we know now that the pyridine is or can act as a base. What is the name of this compound? Pyrene. Furan is O. So if the same thing is this is called furan, and if you don't have these, it's called tetrahydrofuran. So, right? So this thing is a base. Now we know that the basic character is there because these 2 lone pairs are not involved in the bonding. Now, there will be these pyrimidine-like structures will be similar to pyridine. How will they be similar with pyridine? So these will be 6-membered rings. and only one 6-membered ring. So actually they have only one 6-membered ring in their structure, and there are 3 types of such compounds that we are interested in. These are called the cytosine, which has a structure of this. Now tell me whether this nitrogen is involved in, let me just cross check if I got the structure correct or not. Yeah, it's correct. So tell me whether this lone pair is available for bonding or not, or available to react or not. So these are the 3 pyrimidine structures that we will be looking at. Just making sure these are correct. So cytosine and NH. Just for completeness sake, I'm adding this NH in here. Now, the other kind of nucleobase is purine. Now, this purine has a, so purine is a molecule, unlike pyrimidine. Pyrimidine is not, it is based on pyridine, but purine is a class and a compound in itself. So purine is a 6-membered and a 5-membered ring. So this is purine. And there will be, so this is made up of, you can think of this as a 6-membered pyrimidine structure plus a 5-membered imidazole structure. So this is 5-membered, this is a 6-membered. So this is the 6-membered ring, right? And this is the 5-membered ring, right? And we will see there are 2 different kinds of purines that we are interested in. One of them is adenine. Let's draw the structure. adenine. And what is the other one? The guanine. Now, in here, we will be interested about, as we mentioned before. So I'm just removing all these. So any of the information that we will not focus on. So this is a pyrimidine. This is a purine. So we need this information for our next part, in which we already saw the sugar and the phosphate group making this phosphodiester linkage. And now we will have to see how the sugar makes a linkage with this nucleobase. Right? So sugar will come with what? What will it come? And what will the nucleobase come? Think about the amino acid, the peptide linkages. What were similar to that? Not exactly. What do you think the sugar will contribute? What does sugar generally contribute when it is coming together? The OH. What will the nucleobase come? NH. It will come with that NH. There will be an NH-OH bonds happening in here, and we'll look into that. Okay, let's look into okay, so now let's make a nucleoside. Let's make a nucleoside. How will you make a nucleoside? You will take a sugar and then get the nucleobase. So I will give you the sugar. Maybe let me draw it a little bit more clearly so that it becomes easy for you guys. Make a nucleoside with these two things. So what is this? And this is? Make the nucleoside with these two combinations. Because once you know this, you can make any combination, or at least have a feel for what's going on in the structure of DNA, and it won't be a black box. That's what I want. Now to think which bond, which nitrogen is actually coming to do the trick here. This one. So when this and this OH will come together, what will you form? You will form this bond. Okay? The name of this compound is what? This is adenine, right? This is the ribose. But the name is very close to it. What is the name? Adenosine. So this compound is called adenosine. Any doubt?
Is there a carbon and another hydrogen is attached, right, in that sugar? Where the bond is attached.
Where? Here?
Hydrogen, no need to show, but it
Yeah,
is
but it's
hydrogen
there.
is there.
Hydrogen, I'm not showing.
Yeah.
But there is no carbon.
Yeah, carbon is there, but no need to show the hydrogen.
Yeah, I mean, carbon is here. So this ribose is actually making this bond in here, and this compound that you form is called adenine. Now, what I will do is I will make this deoxy ribose by removing this. So what will be the name of this form? What will be the name of this thing? Deoxy-adenosine. Easy?
Yeah.
And you can now do this with all the bonds, with all the bases. We have 4 types of bases. You can do the same with all these bases, right? And so guanine is going to come in with this in here, right? And now I want you to tell me, so this is the part in which the sugar is connecting to the nucleobase and you are forming the nucleoside, but now it is the time to now again bring in what? On this side, the phosphorus, the phosphate group. So when all these 3 things will come together, what will you form? The nucleotide. So now let's look into how can you take this and make into a nucleotide. So I will remove this. I will say nucleotide, but I will have the phosphorus, the phosphate group in here. Tell me how this is going to attach to the phosphate? From which carbons?
Carbon 3 and carbon.
This and this, right? So what is going to happen is, let's look into just from the above. So you will have these PO minus O minus coming in, right? So there will be an OH to start with, and that OH will go away, and you will form this ester bond.
Between O is there. Between phosphorus and carbon.
Yeah. So I'm again making it ribose by calling it OH. So this will be a deoxy-adenosine, sorry, an adenosine. But now you have a phosphate group attached to it. What will you name this? Phosphate. So this compound is called adenosine phosphate. Now you can bring in another phosphate group in here.
Here, that is carbon 3.
Where? Yeah, you can. So there will be another. But I'm not bringing in another phosphate from the 3. I'm bringing it in here. So what will be that? Try.
Between 2 nucleotides connection.
No. So this was a nucleotide. This is already a nucleotide. But there could be different. What I'm saying is, I will not stop at one phosphorus. So see, if there is another. That can come in from here. But at this point of time, I'm not concerned about this. I'm saying bring in another of these things. What will you name this? Diphosphate. And bring in another. This becomes adenosine triphosphate, or heard about this thing, ATP? What is ATP? What is typically called about ATP? This is ATP. So when ATP is produced, we say energy is produced. And when this ATP goes to ADP, we say that your energy was consumed. Just think about it. When we are talking about energy production and consumption, it is just the removal of a phosphate group and addition of a phosphate group. That's what we are talking about. So when you are eating, you are producing energy. Essentially, you are adding a phosphate group to the ADP, and when you are running or sitting here and listening, working, your ADPs or your ATPs are just chipping away these phosphates. That's what we are calling energy. So understanding this structure is important. Right? So this adenosine triphosphate is a part of the DNA, but also a very important molecule in itself. Now, you can have these things and you can create that structure in which, so you have the base and then you have the sugar and then you have this. Maybe I'll just write it this way. Now you know why I made pentagons in here, because it's a ribose sugar, right? This is the part in which you are getting the phosphates attached, and then you have these different bases that are getting attached. Now these bases could be either adenine or guanine or thymine or cytosine. you will have a similar thing going on this side, in which you will have a base and these bases will form either two double bonds or two hydrogen bonds or three hydrogen bonds. Which do you think will be more stable? Two double bonds or two hydrogen bonds or three hydrogen bonds? The three hydrogen bonds. So they will be difficult to break, essentially. Now, we'll just look into how the DNA, how the different bases are just making those three hydrogen bonds or two hydrogen bonds. Okay. So now we have a pretty decent idea about the entire structure of DNA. And when DNA replicates, this one strand and the other strand can actually break apart because they are held together by hydrogen bonds. They are not held together by covalent bonds, right? Because they're easier to break and easier to form. Because if they were covalent bonds, it would be very difficult to actually break them to replicate. So you will not be able to do it. So that is why the hydrogen bonds are important. Now, let's look at the thymine and the adenine structure. Okay. Let me see if I have a easier version of it. Let me draw the different structures once again. So what were the pyrimidines? And cytosine. What are their structures? Their structures are, cytosine is the hydrogen bonding centers in cytosine are this, this, and this. Why? And for this is cytosine and those three. This and this. Similarly, for the purines,